Hello, everyone. I'm Christy Rizk, Senior Editor at GenomeWeb, and I'll be your moderator for today. The title of today's webinar is Identifying COVID-19 Testing, Treatment, and Future Outlook. The presentation today is sponsored by Ovation, a web-based LIMS company purpose-built for molecular diagnostics. Our panelist today is Dr. Bill Massey, Chief Scientific Officer of Vikor Scientific. You may type in a question at any time during the webinar. You can do this through the Q&A panel, which appears on the right side of the webinar screen. And if you look to the tray at the bottom of your window, you'll see a series of widgets to enhance your webinar experience. And with that, I'll turn it over to Dr. Massey. Please go ahead.
Hi. Thanks, everyone. And thank you, GenomeWeb, for giving me the opportunity to share this important information on COVID-19. A little bit about my background. I'm a pharmacologist. I've worked in industry and academia, held academic appointments at Vanderbilt, University of Chicago, the Northwestern University, University of Arkansas, and I'll get to it. So today I'm going to talk about essentially three different major topics regarding COVID-19. First, I'm going to talk a little bit about PCR and antibody testing and how we use that testing in the management of the pandemic. The next phase of the presentation, I'll talk about the role of potential treatments for COVID-19, of future vaccines, and how that plays a role in the management of the pandemic as well. And then lastly, I'm going to talk just a little bit about actions that as a society we can take to help mitigate the impact of any future pandemics. As we all probably know, coronavirus 2019 or COVID-19 is caused by a novel coronavirus, SARS-CoV-2, and first reported in the Chinese city of Wuhan in late December of 2019. This is a very unusual virus. The severity of symptoms varies very widely from asymptomatic patients to severe acute respiratory distress, failure, and death. The virus is most abundant in the lungs, but it's also present in lower levels in the kidneys, the liver, the brain, and the blood, and the heart, and associated with an abnormal immune response. And this abnormal immune response appears in the very severe cases and contributes to the severe pneumonia and respiratory distress that ends up being a major cause of mortality. In fact, you could almost say that the disease appears to be almost biphasic, where initial viral infection occurs and then there's either a turn towards a severe immune response and inflammation and progression of respiratory illness, or some people start to get better. The immune response to COVID-19, it differs from the immune response that's been prompted by previous respiratory viruses. Blancamelo et al. Found that the SARS-CoV-2 virus generally induces very low levels of interferons compared with other respiratory viruses like influenza. And of course, interferons play a role in inhibiting viral replication. In fact, how they got their name, they interfere with viral replication. However, at the same time, it activates cytokines that produce a more general immune response like inflammation, and they're much higher in infected patients than you see in uninfected controls. These results suggest an immune imbalance, where low levels of interferons reduce the cell's ability to limit viral replication, while activation of these less specific immune responses, inflammation responses, actually promotes tissue destruction and pathology. So, taking this into account, targeted immune system treatments may be of great benefit in COVID-19. Now, it's probably likely, very likely, that the number of infected persons is much higher than we see in the number of confirmed cases. The reason being is that confirmed diagnoses require positive testing for the presence of the SARS-CoV-2 virus. And as we know, testing, at least in the United States, has been somewhat limited. It is increasing now, and we're getting more and more prolific at it. But in the initial stages of the disease, of the pandemic, the capacity for testing was limited. So just that alone would kind of indicate that it's likely a much greater number of people have been infected. The other thing is that there's a large portion of asymptomatic cases. There are COVID infections where the patients do not have any symptoms at all. And in fact, it could be as much as 75%. And we really won't understand the true prevalence of the illness until we have widespread testing of these asymptomatic patients. For instance, the Diamond Princess cruise line early in the pandemic, when they tested people there, they found that 40% of the infected cruise ship passengers were asymptomatic and remained asymptomatic throughout the course of the illness. And then there was the aircraft carrier, the USS Theodore Roosevelt, where they had 678 COVID positive sailors, and 60% of them were asymptomatic. In addition to that, early reports from South Korea, where they did widespread testing very early in the pandemic, they estimated up to 80% of patients were either asymptomatic or their symptoms were so mild that they never sought out medical attention. So taking all that into consideration, it's very likely that the number of infected persons is much higher. One of the aspects about this virus that make it spread so rapidly and so fast is that asymptomatic patients can transmit the disease. Strongly suspected anyway. The viral loads, some of the evidence behind that, the viral loads were the same between asymptomatic and symptomatic patients. So viral shedding appeared to be at the same levels. Another thing was that they found in this city in Italy, Italy, they found that 43% of people that were infected with COVID were asymptomatic. So, again, more and more information starts to become available to suggest that a large number of people are asymptomatic and that these asymptomatic patients can transmit the disease, which makes it very dangerous. So asymptomatic and pre-symptomatic transmission can play a role in COVID-19 transmission. And this makes it very difficult to control the disease without strict social distancing because you're spreading it without even knowing that you're out there spreading it. So what can we do to help prevent the spread of COVID-19? One thing we can do is frequently wash our hands because soap appears to destroy the virus very well. If you can't do that, using a high alcohol content hand sanitizer can work as well. Avoid touching the face. And then if you're symptomatic, even if you don't have a diagnosis, quarantining and social distancing appear probably to be one of the most effective methods for prevention of transmission. Now, COVID-19 is very high in its contagiousness. It has an R0 value of greater than 2, which essentially means that each infected person infects more than 2 others. And I would say that because of the high number of asymptomatic cases, it may even be greater than that. If the R0 value is reduced to less than 1, then of course the disease will subside because you're not spreading it, each person's not spreading it to someone else. Now, COVID-19 testing plays a huge role in how we manage this pandemic. Using testing, we can help reduce these deaths by identifying the cases and then allowing us to take the appropriate actions in those patients that are positive in order to slow the spread of the virus, such as quarantining. As the cities and the states, in the United States anyway, lift their stay-at-home and physical distancing orders, we expect that there will be a big spike in the number of infections because of asymptomatic spread and interacting with a lot of people. The FDA has granted emergency use authorizations for a number of diagnostic tests that are used to detect the coronavirus. And companies and universities around the world continue to develop new ones. The two types of tests, the main types of tests that are available to detect COVID-19 are viral tests, which actually detect whether or not you have a current viral infection, and antibody tests, which determines whether or not you've had a previous infection or exposure. So when you look at a PCR-based test for viral testing, what you're doing, you're detecting current infection by detection of specific nucleic acid sequences for the nucleic acids that code for virus-specific proteins. At Vikor Scientific, we test for 3 different proteins that are specific for COVID-19 and very selective sequences that don't undergo a lot of mutation. These areas of RNA don't undergo a lot of mutation. And so, therefore, there's more likely to be accurate throughout the pandemic. We test for the M protein, the S protein, and the ORF1ab protein. PCR testing is very, very accurate. It's greater than 99% accurate. However, there are a number of challenges in PCR testing that seem to be specific for the COVID virus. One thing is that it can take up to 14 days after infection to actually test positive for COVID-19, even though you're already infected. And of course, during that 14 days, if you're symptomatic, maybe you're staying at home, but if you're not, you're out there spreading the disease to other people. And again, pre-symptomatic patients are assumed to be able to transmit the disease just like asymptomatic patients. And there seems to be a phenomenon where people continue to be testing positive over a prolonged period of time, even over a couple of months. Now, some of the issues with that may be the detection of viral fragments. In other words, not live virus, but viral fragments that we can detect with the PCR testing. And some of the evidence behind that being the case is that they've taken samples, swabs of people, and tried to culture the virus in these patients that were being positive for a very long period of time. And they were unable to get the virus to grow. So that suggests that the viral fragments that are being detected are not live virus and are not viable, and thus viral fragments are left over from the infection. Another phenomena that we've seen, especially in some of the early hydroxychloroquine studies that were conducted in, published in April and even available in late March, were that patients would be tested every day in these studies examining the effect of hydroxychloroquine, see whether there was a potential therapeutic effect. And even though they were tested every day, some days the test would come up negative, and then the next day it would be positive. And so you had this alternating positive and negative test result phenomena, and really don't understand, and no one really understands why that might be the case. Usually with a negative test with PCR, it's because you didn't get a sample, because you didn't get enough sample in order to detect it. And so it's a sampling error, but it appears to be very common. The other thing that we need to look at is the antibody production from COVID-19. The good thing is that almost everybody who recovers from COVID-19 produces antibodies against this new virus. So a study at Mount Sinai by Weinberg and his associates found that more than 99% of people that were infected eventually developed antibodies to COVID-19. And age and sex had no effect on antibody development. That's very good news. And it's very strongly suggested that they are immune from reinfection, at least for some period of time. Though how long that is, we don't know. And one of the reasons that I say that is that other studies have shown that in macaques that you can give the monkeys the virus, they'll produce neutralizing antibodies, which actually attack and inactivate the virus. And these neutralizing antibodies prevented these monkeys from a direct challenge of reinfection later. So that, that's very strong evidence that perhaps this immunity is developed to COVID-19. The other thing is we haven't seen a lot of cases, I haven't seen any cases I'm aware of, where someone was actually cured from the virus and then got reinfected at a later date. So it appears that for the most part anyway, that people do develop immunity after infection with COVID-19. Now, the interesting thing about that study is that it takes sometimes up to a month to develop these antibodies after the initial symptom onset. And then, of course, like we had talked about earlier with PCR testing, that some of these volunteers, 20%, tested positive for COVID-19 virus for 2 or more weeks after their symptoms had ended. So in other words, that's a very consistent picture that's showing up around testing. So the antibody testing that's being conducted now detects antibodies to the virus that were produced when someone got infected as an immune response to the infection. And again, it can take 1 to 4 weeks after infection to make antibodies, which means that during that time period, you can get a false negative result if an infection was just very recent. So again, that's another one of the issues we have to take into account when we try to make inference from COVID-19 test results. Now, having antibodies to the virus probably protects you from getting that virus again, but we don't know how long that protection might last. So again, just because you got infected doesn't mean you can't catch it again sometime down further in time. We just don't know how long that period of protection might last. And so anyway, these the delay in getting developing the antibodies, and then there's been also a pretty high rate of false test results with some of the antibody testing. So these are issues which make it a more cloudy picture as far as testing for previous exposure to SARS-CoV. But the good thing is that we do produce antibodies. Another good thing is that those antibodies appear to be neutralizing antibodies. And if that's the case, it should give us some immune protection from COVID-19. But we need to understand how many people have had the illness in order to understand whether or not we're able to get to a place where we have herd immunity, which usually takes about 2/3 of the population to carry antibodies to the virus in order to have that sort of herd immunity. One of the tools that appears to be very useful in managing the pandemic and the spread of the virus is social distancing. So if we can slow down this transmission rate, hopefully that gives us a break from the healthcare strain on our system and emergency rooms and ICU units that are getting, overcrowded due to severe cases. It does appear that social distancing works. Some researchers in France at the Pasteur Institute modeled their coronavirus outbreak and found that their lockdown reduced viral spread by 77%. Now, this is a modeling study, but that's still a pretty big effect. And then also, there's new evidence in China that show that school closures, travel bans, and these other painful methods have been effective in curbing the coronavirus epidemic. And of course, when we start to relax these social distancing guidelines, if we see a resurgence in the infection rate, then again, that will be evidence that social distancing is effective. There was another Chinese study that showed that if you were able to get the infections detected quickly and isolate those people, that was the most effective step that we could take in order to limit the spread of COVID-19. The study estimated that if we would have done if China would have instituted these actions earlier, only 3 weeks earlier, that 18 times, if they would have instituted these 3 weeks later, that 18 times the number of people would have been infected. So that's a huge impact on the number of people. And they also estimated that after a stringent lockdown, that there were essentially 15 to 20 encounters per day between people where you have an opportunity to transmit the virus were reduced to roughly 2 per day. And again, the reduction of transmission will bring the epidemic under control. So this huge pandemic and unprecedented health emergency that we're undergoing prompted a magnitude of response, a huge magnitude response to developing treatments and vaccines to the coronavirus. A huge global effort essentially is underway. The Milken Institute lists currently, and it changes from day to day, 223 treatments under investigation and 141 vaccines that are in development. And it may take some time to develop a vaccine. There are some good signs out there, and I'll talk about those. there are existing drugs that may be successfully repurposed for use in the treatment of COVID-19. Now, an effective treatment in the absence of a vaccine may be the best weapon that we have against this current pandemic. There was a lot of talk early in the pandemic about hydroxychloroquine and chloroquine, which were anti-malarial drugs that have been around for a long time. And there was a study out of France that showed that people got better and faster, and that there was a mortality benefit, things like that. That study has come under a lot of criticism, so I don't really want to put a lot on that study. But in light of the emergency that we were facing, the FDA decided to grant the emergency use authorization for hydroxychloroquine and chloroquine. The clinical results, though, in subsequent studies have been mixed at best. For instance, there's some issue with hydroxychloroquine regarding QT prolongation and potential for arrhythmias, cardiac arrhythmias, and the death and mortality associated with that. And that's one issue. And then also azithromycin. If you add azithromycin to that, azithromycin also causes QT prolongation. And again, the two things together can increase your risk of developing a heart arrhythmia. A study recently published in Lancet showed that patients receiving these hydroxychloroquine or chloroquine were dying at higher rates than patients with COVID-19 who weren't receiving those drugs. And then there was another study published in JAMA which showed that hydroxychloroquine, with or without azithromycin, didn't appear to help people with COVID-19. And then actually yesterday, there was a study released that showed from Minnesota that showed that hydroxychloroquine did not have a preventative effect on COVID-19. So there's a lot of data that's come out that's kind of dampened enthusiasm around hydroxychloroquine and chloroquine. The next drug I want to talk about that has potential, and I think it has great potential, is remdesivir. And remdesivir is a antiviral drug that's made by Gilead, and it's currently an experimental antiviral drug. It inhibits the virus by mimicking the building block of the coronavirus's genetic material, RNA. And so when the virus starts to copy its RNA, the remdesivir takes the place of some of these building blocks. And of course, that results in a faulty replication and slows the virus up or stops it. On April 29th, it was announced that remdesivir shortened recovery time for seriously ill patients with COVID-19 by 4 days in a clinical trial comparing the drug with placebo. Now, that might not seem like a lot, but in the current situation, it was very promising. And it was considered that it was so promising that it was considered evidence for the Safety Oversight Committee that was overseeing the study to stop the clinical trial in order to give everyone in the study the remdesivir. Now, it's important to note that remdesivir did not have a statistically meaningful effect on mortality. 8%, fewer people died on remdesivir versus the placebo group, but it wasn't that great of a difference. It wasn't statistically meaningful. One of the issues with these antiviral therapies is when do you give it in the course of the illness? If you wait too long and the virus gets established very well, it may become less effective from a clinical standpoint in relieving the symptomatology and changing the course of the disease. So there appears to be evidence that if you give remdesivir earlier in the course of the illness, that it can have a bigger effect than if you give it later. A study in China showed that if you give it within 10 days of symptom onset, it sped up the recovery versus waiting later. The previous results from studies of patients given remdesivir for compassionate use, because there was no clinical trial, this is just anecdotal, showed that 36 of 53 people that were given the drug needed less supplemental oxygen afterward. And this was reported in the New England Journal of Medicine, April 10th. And based on all of these studies coming out with remdesivir, and also anecdotal reports where we've had some very dramatic improvements in patients, the Food and Drug Administration issued emergency use authorization for the use of remdesivir in COVID-19 patients who were hospitalized and whose blood oxygen levels had dropped to 94% or lower. One of the interesting things is that Gilead ran a clinical study, and they found that that treating for 5 days instead of 10 may be enough. So originally the course of therapy was for 10 days, but Gilead showed that 5 days is enough. And so currently, if we were able to do that, we could double the number of people we could treat with available drug. So, that's a good thing because we can double the number of people that receive remdesivir when they need it. Gilead is scaling up the production, and they think that they can make 1 million treatment courses by the end of the year. And of course, if you use a 5-day treatment instead of a 10-day treatment, you expand that to 2 million treatment courses. So other antivirals that are under investigation, there's a combination of interferon beta 1b, lopinavir, ritonavir, which is also named Coletra. It's marketed as a branded combination, and ribavirin, have shown that if you take that, there was a shortening of the disease course from 7 days versus 12 days from when they just used Coletra, lopinavir, and ritonavir alone. So 7 days versus 12 days when you added the 3-drug combination versus just Coletra alone. And the other thing that was very promising is that it reduced the amount of coronavirus, the viral load in these patients. And this was published in the Lancet. Now, Coletra alone, it kind of has disappointing results. And this next drug I'm going to talk about, lopinavir, showed that it was, not as effective as lopinavir, which is a antiviral from Russia. It's licensed in Russia and China that they use for prophylaxis for influenza. The good thing is it appears to have broad spectrum antiviral properties. And so if you look at the study that they used comparing it, they found that it was more effective at reducing viral loads. So reducing viral loads means it has an antiviral effect. There was another trial that hadn't been published yet that found that neither treatment were effective in improving outcomes in mild to moderate COVID-19. There's a Japanese drug, favipiravir. This drug is approved in some countries, not in the United States, but in some countries for the treatment of influenza. And there are some published reports, unpublished reports from China that suggest that it may be an effective treatment for COVID-19. Sorrento Therapeutics got quite a bit of a splash in the media by announcing that they'd found an antibody that may actually, they used the word cure for COVID-19. I'm a little more conservative about that, but I think an antibody to the virus is a very directed, targeted therapy that has a lot of promise for being effective. So what happened was Sorrento were screening numerous antibodies in its library, and they discovered hundreds of candidates that bound a particular subunit of the SARS-CoV-2 spike protein, the spike protein being the protein that the virus uses to attach to the cells. So the idea is that if you can block that spike protein, then you can block the virus from being able to attach and infect the cell. So in mid-May, Sorrento announced that they had a candidate out of all these antibodies that they had looked at, STI-1499, that could completely block COVID-19 infection in vitro. Now, such a drug would be useful not only to treat COVID-19, but it could also serve to help prevent infection by preventing the virus from ever getting a hold. So in someone who is at high risk, you could possibly use this as a preventative. And the company is going to produce an antibody cocktail with multiple antibodies in it that would protect from COVID-19 and hopefully would remain effective because you look at multiple targets, even if the virus undergoes mutations. And so, having multiple targets increases the chance that that antibody would be effective over time and through mutation. So there are other monoclonal antibodies that are out there in development as well. Vir Biotechnology, again, isolated antibodies from people who had survived SARS. And they're working with the Chinese firm Wuxi Biologics, and they're going to test them as treatment for COVID-19. AbCellera identified potent antibodies as well, recovered from patients, convalescent plasma patients. In partnership with Eli Lilly, they just entered that partnership. They've started their clinical trials. And the good thing is that they think they can accelerate the development very rapidly and maybe even release this in the fall of 2020, which is very good news. So if we can get something prior to the estimated resurgence that's supposed to occur in the fall, the second wave that's going to occur in the fall, then that would be a very effective treatment, very valuable treatment. The previous drug I was talking about, the antibody, is isolated from people who've already had the disease. And so when you take the blood out, you fraction it out, the plasma portion has the antibodies in it. And so if you take plasma from blood from patients that have had disease previously and then separate it and get the plasma, they call that convalescent plasma. And there's data from Italy that in that plasma, of course, would have the antibodies in it. And there's data from Italy that indicates that this convalescent plasma has reduced COVID-19 mortality when it's given to a patient as a drug. So far, there's been 12,000 COVID-19 patients that have received convalescent plasma. And the safety of that has been established. And so now we're looking at efficacy to see whether or not it actually works in combating the virus. In the U.S., the National COVID-19 Convalescent Plasma Project, it's a multi-center project consortium, is evaluating the use of convalescent plasma. And hopefully we will have efficacy data within the next month. Another approach to dealing with COVID-19 is to try to dampen the immune dysfunction and what they call the cytokine storm, where you have all these immune signaling molecules that are released and cause this huge overactive immune response, which does damage. It causes the pneumonia in the lungs and damages the respiratory epithelium. And so if we can suppress that immune response, that inflammatory response, we may could have a very effective treatment for prevention of the more serious sequelae associated with coronavirus, which are the acute respiratory distress syndrome, leading to death in a lot of patients. So if we can offset that cytokine storm, we can prevent people from having to use a ventilator. And using a ventilator can cause damage in and of itself. It can cause fibrosis of the lung because of the mechanical ventilation that occurs. So if we can reduce the need for the ventilator, reduce the cytokine storm, perhaps we can reduce the pneumonia and reduce the mortality of the illness. So one of the issues that we have, though, that's a global issue with anti-inflammatory and immunosuppressant drugs is that it's kind of a double-edged sword. So you want to dampen the inflammation in that inflammation response, but you don't want to suppress the immune response, which helps you to clear and get rid of the virus. So, the deal is that we have to figure out how to suppress that inflammatory aspect of the infection without damaging the ability of the immune response to clear and eventually get rid of the virus. So Along with the efficacy, safety is one of the main concerns that we have because, again, if we dampen the immune response, then we're going to have real trouble in being able to clear it eventually. So you want to get the inflammation, but not hamper the ability of the body to clear it through the immune response. Now, that means that we really need narrowly targeted agents so that they don't hit so many of the different immune pathways and deal specifically with the inflammation. And of course, the question though is, which are the best targets? We use corticosteroids. They're well studied, been around a long, long time, but they're also the bluntest tool for muting inflammation in the immune system. So again, with corticosteroids, you have that issue of, I do want to get rid of the inflammation, but I'm also suppressing the body's ability to fight the virus. And there are a number of clinical trials that are ongoing looking at that. So as far as targeted drugs, there are several immune suppressants that are being targeted to see, again, whether you can quell this cytokine storm and reduce the progression to acute respiratory distress syndrome in these COVID patients. Baricitinib is a Janus kinase inhibitor. It's a drug that's been used in rheumatoid arthritis. And it has a potential antiviral effects by its inhibition of viral endocytosis. However, it appears that it can also block the antiviral effects of the interferons, which are some of them are mediated through that Janus kinase signaling pathway. So again, it's selective, but there is a concern about it because it could actually interfere with the production of interferons, the effective interferons that are mediated through that Janus kinase pathway. And then there are interleukin 6 inhibitors that are being developed that are specific for interleukin 6 that may have very targeted anti-inflammatory activity. In addition to that, FDA has also approved a device that actually filters the blood for cytokines and remove those from the blood and reduce the inflammation produced by it. So interleukin 6 antagonists, very targeted immunoinflammation suppressant therapy has been studied, and there are several things being looked at, the monoclonal antibodies for interleukin 6. And so in a retrospective study of 200 patients, COVID-19 patients, they experienced very severe illness, had very high levels of the inflammatory cytokine interleukin 6. Now, the interleukin 6 is important to target because it contributes to the cytokine release syndrome, this cytokine storm, which is primarily the cause for the exacerbation of these severe reactions to the virus and lead to the acute respiratory distress syndrome, even when the viral loads are starting to diminish. Again, that second phase of the illness. There are tocilizumab and saralimumab, who are monoclonal antibody antagonists that are being used to treat rheumatoid arthritis. And some of the early results with those appear to be promising. The published data is not yet forthcoming, but we expect it soon. And again, because of how tightly it targets IL-6 and not so much other aspects of the immune function, it may actually reduce the side effects that we don't want with immune suppression therapy and lead to a very beneficial anti-inflammatory response. There was a phase 2 study of saralimumab that showed that there were some positive trends in these patient groups that were critical, but negative results in patients in severe group. And by severe, they mean they weren't yet intubated, but they needed oxygen supplementation. So again, we need to understand exactly when in the course of the disease to try to interfere with these therapeutic agents in order to have the biggest effect on progression of symptomatology. And because there's such a wide variety of symptomatology in COVID-19 patients, there may be this subset of COVID-19 patients that have that exaggerated inflammatory response, a cytokine storm, that may be of most benefit to these drugs. There's a promising new drug target, and again, this is a new drug target, and it'll take some time to develop this drug, but it may have a lot of utility in the future against other coronaviruses, because it's not so specific for SARS-CoV-2, but would be more of a general coronavirus treatment. It's called M-protease. M-pro, for short, is a key enzyme that's involved in the viral replication and transcription. And so that makes it, if you can stop the viral replication, you can essentially have an antiviral effect. So it makes it a very attractive drug target for SARS-CoV-2. It's also very specific for coronaviruses, so there's little risk of off-target side effects. So inhibition screening for inhibitors that fit in this groove of the M-pro enzyme have shown that they are very powerful at inhibiting this virus. And there are some that actually have had Epsilon that have safety data from humans. So they're a little bit further down the road. Again, by inhibiting the enzyme, they inhibit viral replication. And again, because this is so specific for SARS-CoV, these other coronaviruses also have this same site, a binding site. It raises hopes that this single compound might be useful across a number of different coronaviruses. Now, I'm going to go through a list of other treatments. It doesn't mean to be exhaustive or comprehensive because there are so many, but these are ones that may have an impact soon. Atherosis and mesoblast have already started studies looking at stem cell treatments that seem to be a benefit in the acute respiratory distress syndrome by reducing the amount of fluid that accumulates in the alveoli and, again, reducing pneumonia and the respiratory distress syndrome. Again, just starting phase 2, so we don't really have any efficacy data yet. Nafamostat and Chemostat are serine protease inhibitors that are found that they block the entry of the SARS-CoV-2 virus into the cells. Nafamostat seems to be better at that, 15-fold higher efficacy than that of Chemostat. These are also currently just entering phase 2-3 trials for efficacy, so no efficacy data yet. Nitazoxanide is a anti-infective, actually not an antiviral per se, but a broad-spectrum anti-infective that has efficacy in both parasitic, bacterial, and viral infections. In viral infections, such as the infection with the MERS-CoV, the Middle Eastern Respiratory Syndrome, it acts by blocking the maturation of the viral nucleocapsid N protein, and then that promotes the production of the particles. So by inhibiting that, it prevents the production of viral particles. And this drug is being tested in combination and against hydroxychloroquine and in combination with another antiparasitic, ivermectin. Now, ivermectin is a drug that's been around a long time, broad-spectrum antiparasitic. It's thought to work in the COVID-19 virus by binding and destabilizing cell transport proteins. And the virus uses those to enter the nucleus so it can start to replicate itself and produce more virus, and the viral replication leads to progression of infection. So in a 1,400 patient observational study, they showed that ivermectin was associated with a lower death rate. 1/3 the amount that was in the control group that didn't get ivermectin, and shorter hospital stays. Also, fewer intubated patients, these are really seriously ill patients, died in the ivermectin group as well. So several trials are currently underway, and we'll see whether or not this potential efficacy of ivermectin continues to hold. Bevacizumab And a monoclonal antibody is directed against VEGF. That's vascular endothelial growth factor. Is used in a variety of different cancer treatment to stop the vascularization of tumors or they get fed. So by suppressing that, it reduces the blood flow to these tumors. But also by suppressing VEGF, you can reduce vascular permeability. And so when the permeability of the vasculature in the respiratory epithelium is increased, that's when the fluid leaks out and gets into the alveoli, causing the pneumonia that is such a problem in these COVID-19 patients. So if you can inhibit that, then you can potentially inhibit the accumulation of fluid in the lungs for those that are progressed all the way to the acute respiratory distress syndrome. Another drug which has been around a long time, famotidine, is a histamine 2 receptor blocker it's used as a antacid for gastrointestinal disorders, GERD, and things like heartburn, things like that. There's very little evidence, but it got a lot of press because it's a very cheap drug. It's a small molecule. It's easy to make. You can get lots of it. It's very available. The evidence actually came from a Chinese study where they showed that people that were on, not on proton pump inhibitors, but on the cheaper H2 blockers did better with COVID-19. However, we don't know. There are so many other things that could be causative in such a observation. Like, for instance, maybe the people on the cheaper drugs had lower health status because of poverty. There's a lot of things that could be that could obscure the accurate interpretation of that data. So the evidence on that's pretty sketchy, and I don't really know of much evidence that would indicate famotidine would be effective in the COVID-19. Probably the best ultimate treatment for COVID-19 would be preventing infection in the first place through a vaccine. So vaccines protect people by training your immune system to produce antibodies to recognize and attack the virus whenever they're encountered. So if you can develop these antibodies, just like if you become infected and you survived it, you develop antibodies that way. If you can give a vaccine, which usually is a protein that's specific for the virus, so that you just inject the protein, or you inject an inactivated virus or dead virus into someone, your immune system sees that as foreign, and it creates antibodies to many different sites on the virus, and then you develop immunity from the antibody load that you carry. Now, the great thing about vaccines is not only they protect the person who's vaccinated, but they also protect the community around the person, because if you have a number of people that are vaccinated, then you can have what they call herd immunity, where enough people in the population are immune, either through previous exposure or through vaccination, are immune to the virus, then the transmission is interrupted. So, and if you're vaccinated, you can't pass the virus on to others because you can't catch it yourself. And again, this herd immunity is what we desire whenever we engage in a vaccine campaign. There are many, many projects underway, and the results so far have been promising, and we'll talk about a few of them. Moderna is testing a messenger RNA vaccine, which is a very unique and new technology, which is very important, I think, not just for this, but in COVID-19, but for the future of a lot of other conditions as well. In short, what they do is they administer messenger RNA that codes for a protein, and that messenger RNA is encoded in lipid nanoparticles, and it gets into the nucleus. Your cells make the protein, and then you develop antibodies to the protein that's produced in through the messenger RNA. And so the good thing is that Moderna's messenger RNA vaccine is producing antibodies in the 45 trial participants, and that they've commenced a phase 2 study. Oxford University has a modified virus that they're using a vaccine, and they've shown that it was effective in rhesus monkeys, and the rhesus monkeys were immune to a direct viral challenge. So that indicates that not only were they producing antibodies, but they were neutralizing antibodies. And they're starting a study. This gets already started, and hopefully we'll have drug available in the fall. That's very ambitious, but they're very optimistic. They've partnered with AstraZeneca, which has already started to manufacture the vaccine, so that as soon as it becomes available, they can distribute the vaccine worldwide. There are other vaccines that are in development, and Pfizer's developing another messenger RNA vaccine. I'm going to go a little faster due to time. And I also wanted to talk a little bit about a study that I conducted at Vikor Scientific. What we did was we looked at, we had 12,075 patient samples come into the lab between March 25th and April 22nd. And what we did, we ran, not only did we test them for COVID-19, but we tested them for 40 other known respiratory pathogens, both bacterial and viral. And the reason we did this was because co-infection or subsequent bacterial infections have shown to have been a big contributor to morbidity and mortality in previous respiratory viral pandemics, including the 1918 influenza pandemic, as well as the Hong Kong flu in 1968. So we wanted to see whether or not people were co-infected more frequently with COVID. In other words, the COVID positive patients, did they have co-infectious pathogens more frequently than those people tested that were COVID negative? And so what we found was that we had 1,690 COVID positive patients. We didn't find any differences in COVID infection by race, by gender, or sex. We did, however, find that advanced age, and even if you were in a nursing home or residential facility, that did increase your rate of COVID infection. And in the nursing home, it increased it even when you controlled for the advanced age. So this data has been submitted for publication, so hopefully we'll get a result on that soon. What we found was that the COVID-19 positive patients had a significantly greater prevalence of co-infection with potentially very nasty respiratory pathogens like Staph aureus, M. catarrhalis, and Klebsiella pneumoniae. These pathogens can in and of themselves cause pneumonia. So the fact that COVID positive patients had a higher rate of them could be due to a couple different things. One, it could mean that COVID positive patients are more susceptible to these pathogens. It could also mean that the people already had these pathogens colonized in the respiratory tract and made them more susceptible to COVID-19. I don't know which one, because we only tested one time point and they were co-infections at that time point. But anyway, this is very important, because if you don't test for co-pathogens with COVID-19, you might miss a treatable bacterial pathogen that could produce symptomatology very similar to COVID-19 as far as respiratory symptomatology of pneumonia and respiratory distress. So if you know that you have them, then you can direct treatments to them and possibly improve a subset of patients that have COVID-19 co-infection with other pathogens. So again, we saw that the elderly group had double the rate of that of younger patients, and that residential patients had higher rates even when you controlled for age. And we also found that co-infections were more prevalent. Some of the co-infected pathogens, again, were nasty pathogens that cause pneumonia. And these co-infection rates, the co-infection rates also, were disproportionately increased in the elderly and residential patients. So it's important that we look for other co-infected pathogens in order to treat those that are treatable and relieve those symptoms that may be due to the co-infected pathogen. So preparing for the next pandemic, we live in a connected world. We have things coming from everywhere and people interacting between the continents. And this pace of globalization, urbanization, and mobility is definitely contributing to our susceptibility to pandemics. Again, migration, expansion of urban areas into previously natural habitats increase our proximity to animals and other potential sources of new viral pathogens. And again, they also, by proximity, increase the likelihood of spreading. And more diseases are appearing about which we have little or no knowledge, just like coronavirus 19, COVID-19. There are, we have these known threats as well, and these pandemics are going to be inevitable. We're going to have another one. It's one thing, a constant throughout human history. But even though we knew about this, we still weren't prepared. And I'll give us a break because the magnitude of this current pandemic might make it difficult to be completely prepared for it in any way. Now, hopefully we will learn from our current pandemic. One thing that we know is that we need to have sufficient stockpiles of personal protective equipment. And we need to have systems in place to coordinate the distribution and our response to these pandemics. We need stronger healthcare networks, and we need coordination tools. Emergency operations centers, these are hubs where government and public health organizations can review data and help formulate strategies. And we need to have systems that can quickly identify and contain these outbreaks like we had with the World Health Organization. And we need to strengthen those collaborations between countries and between organizations in order to effectively deal with future pandemics. And again, we need to actively monitor for new pathogens by looking for mutations in known human viral pathogens. So 4 different things that the Gates Foundation says we need to concentrate on. One is progress in research and development. And I think this is one place where we've actually shown, where we really shine in this current pandemic, is the massive mobilization of resources to develop new therapeutics and vaccines. Well, that's important. We need to be able to track disease. By knowing where it is, we can help isolate and we can help to contain these better. And through collaboration between governments and other public health organizations so that we can share these resources and information. And of course, we're going to have to get more proactive about funding and investing in all of these areas. And we know now what the great price is in paying for this after the fact. Anyway, that's the end of my webinar, and I'd like to thank you for your attention, and we'll open it up for questions. I also included a slide with the references that I referenced in the talk so that you can delve into these subjects more deeply. Thank you.
Great, thank you, Dr. Massey. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. And before we begin the Q&A, we'd like to ask attendees to take a moment after the webinar has ended to give us feedback by taking our exit survey. All right, we have a long list of questions, so let's dive right in. So Bill, is IL-1 induced during infection, and are cytokines processed by inflammasome major players in the inflammatory syndromes caused by infection?
So essentially, usually, interferons are generated by viral infection as our natural defense to interfere with viral replication. Interleukins as well, but those primarily, they can actually interfere with the interferon. So IL-6 is a primarily inflammatory signaling molecule. So by targeting IL-6, we hope that we can target the inflammation response without dampening the immune response to it. So that's to me is why IL-6 seems to be a very promising target.
And there has been some data that shows that post-infection antibody levels can be different, and that the antibodies produced can differ. So do asymptomatic subjects also produce significant levels of antibody protection? And could this be used as a source of antibodies for treatment?
Yeah, that's a good question. There doesn't appear to be a difference in the amount of virus that's carried and replicated in asymptomatic patients versus symptomatic patients. Therefore, one would expect that there would be no difference in the level of antibody development with asymptomatic patients versus symptomatic. So there's no evidence to the contrary that asymptomatic patients would be a source of antibodies. That they would have immunity, and that immunity would be neutralizing antibodies and be effective in preventing future infection. So That's a good question, and it appears that there's really no difference there, even though I don't think anybody's ever done a controlled study to look at that. But the viral loads are identical. When you swab people that are asymptomatic, you get the same level of virus as you do when people are symptomatic. So.
There's also been some research lately that shows that COVID-19 could be a vascular disease and not just a respiratory disease. Does that change the outlook on potential treatments?
Yes. You know, as you know, the ACE2 receptor, the angiotensin converting enzyme receptor 2, is the binding site that COVID binds its spike proteins to. And those are found throughout the circulatory system, in the heart muscle, in the vascular epithelium. And as we know, we know that that enzyme actually breaks down angiotensin 2 into inactive molecules. So by that enzyme, angiotensin 2, if it accumulates, can be dangerous in and of itself. So yes, and then we have the clotting, the clotting issue that we've seen with COVID-19, where patients are having blood clots form. And I think there's a combination of factors. I think there's a direct effect of the virus, but I also think that in these patients that are developing these, they're immobile, they're prone, they're not moving around. And again, that in and of itself is a risk factor for developing blood clots. So I think that, yes, I think that damage to the heart muscle is very possible, probably been seen. And we're so early that we really don't have a lot of controlled data to back up these assumptions, which are based on, biology, known biology of these systems. But I think that, yes, cardiovascular sequelae are very important to look for and to monitor for in COVID-19 patients.
Great. Thank you, Bill. I'm afraid that that's all the time we have for today, so we'll have to wrap up there. We'd like to thank Bill Massey and our sponsor, Ovation. If we didn't have time to get to your question, we will be following those up with Dr. Massey. As a reminder, please look out for the pop-up survey after you log out to provide your feedback. If you missed any part of this webinar or wish to listen to it again, a link to an archived version will be emailed to all attendees. Thank you for joining us for this Genome webinar.